Hello and welcome to the podcast Byzantium and Friends. This is part two of a series of roundtables, two of them, that were organized by Tina Sessa and Tim Newfield, with the least clickbaity titles that you're going to see on a podcast. Anyway, and the last one was introduced by Tina, and so today's discussion is going to be introduced by Tim very briefly. The previous roundtable was on ancient human DNA and its historical implications, and today we're going to be talking about pathogen DNA. So, Tim, welcome to the podcast. Tell us a little bit about this roundtable discussion.
It's wonderful to be here. Thank you, Anthony, very much. So this is the second of two podcasts associated with a special issue of the journal Studies in Late Antiquity that Tina Sessa, cultural historian of late antiquity, and I are co-editing. I'm a disease and environmental historian of the same period. The special issue focuses on the paleosciences, think paleogenetics, paleoclimatology, all the paleos, paleoseismology, pollinology, paleoclimatology, if I didn't already say that. The special issue is meant to introduce scholars of late antiquity to the natural sciences of the human past, to the ways in which scholars already active in that field are engaging with the natural sciences, and to the debates that are playing out in that subfield. Footnoted and slightly tweaked versions of these podcasts will appear in the special issue. And as you noted, the first podcast focused on human paleogenetics and migration, but this one focuses on pathogen paleogenetics. We focus to some degree on first pandemic plague, but plague gets possibly too much attention, at least in my opinion. And here we speak a little bit more broadly about infectious disease in the late antique period and the challenges that interdisciplinary teams engaged in the disease history of the era are tackling.
Thank you, Tim. And if I might say a word about the background of this conversation, it was deemed necessary in part because the methodologies and objectives of two groups of scholars who are studying ancient pathogens and historical diseases have diverged significantly enough that we now need to think about how to integrate them. And I'm referring specifically to the scientists who work with ancient DNA, like in the lab or through a lab, and historians who work primarily with written sources. Now, there was a time, not a few decades ago, when there was only the latter group, and speculation about historical diseases and pathogens was very impressionistic. You look at a text, you look at the symptoms that are reported, you kind of like puzzle it a bit, and you say, well, that looks a little bit like this to me. And it was all considered very speculative and not very sound at all correctly. But over time, we've now begun to access ancient DNA, both human and of pathogens, and this has opened up a whole different picture of the history of ancient diseases. And we are now at a kind of crisis point where we don't exactly know how to integrate what the lab is telling us with what our written sources are telling us. Often, scholars on either side of this divide aren't entirely aware or not trained in the methodologies used by scholars on the other side. All right. A couple of notes before we get started. Some of the participants refer to Yersinia pestis. This is the bacterium that lies behind the Black Death and the Justinianic plague. This is known to everyone in the conversation, but may not be known to everyone in the audience listening to this now. Also, I will not be moderating this conversation. That was done by Dionysios Stathakopoulos, a professor on the University of Cyprus. I was there, though, and kind of giving stage directions silently about how to organize a podcast. So I had fun doing that, but you won't be hearing from me after this point. One more note. One of the contributors is Kyle Harper, professor at the University of Oklahoma. His mic cut out just at the moment where he was saying his last name when he introduces himself at the beginning of this discussion. So I wanted to make a point of stating it. Kyle Harper, author of the best-selling book, The Fate of Rome. Excellent. Let's get right to it. with thanks to medievalist.net for reposting these episodes on their website.
This is a discussion that will form part of a special issue of Studies in Late Antiquity on paleoscience and the study of late antiquity. It is meant to be an open and frank discussion between different disciplines in order to illuminate both the advances in the field as well as the challenges that we all face. But first, let everyone briefly introduce themselves. Tim?
My name is Tim Newfield, and I'm a historian of disease, or a historical epidemiologist with a focus on the pre-modern period, and even more so on the late antique Mediterranean.
Tina?
Hello, everyone. My name is Tina Sessa. I am an historian at The Ohio State University of Late Antiquity, and I am an expert in cultural and social history.
Maria?
Hello, my name is Maria Spyrou. I work at the Institute for Archaeological Sciences at the University of Tübingen in Germany. I'm an archaeogeneticist specializing in ancient pathogen genomics.
Marcel?
Hi, I'm Marcel Keller. I'm a postdoctoral researcher both at the University of Bern in Switzerland and the University of Tartu in Estonia, and I'm also working on ancient pathogen genomics.
Kyle?
Hi, I'm Kyle, historian at the University of Oklahoma, and I work on the late antique period and the intersection of human history and the natural sciences.
And I am Dionysios Stathakopoulos. I teach at the University of Cyprus, and I'm a social historian of the Byzantine world. We will begin this discussion by defining the state of the field. So my question to Maria and then Kyle is, what are the major advances made in interdisciplinary pre-modern disease studies over the last decade? Where are we and how did we get here? What obstacles have been overcome and what obstacles remain? Maria.
Thank you. Speaking from the ancient DNA perspective, I think that the introduction of ancient genomics and perhaps more importantly and more recently, ancient metagenomics has been a quite useful tool in our understanding of pre-modern infectious disease. So for example, over the last 10 years, we have gone from using ancient DNA as a diagnostic tool with just very small data sets. And again, just to give an example, in 2013, so 10 years ago, we only had very few studies describing pre-modern pathogens on the genomic level. For example, the famous Black Death paper from 2011 or the leprosy paper from 2013, and a paper describing hepatitis B virus that many of us often forget to mention. And we have now come to a phase where we have more than 600 available ancient microbial genomes that have been published. So we're now in a position where we can actually produce much larger data sets, and we have made important steps in understanding and learning how to deal with the complexity of these data sets, to really discussing more about sort of the intertwined histories of infectious diseases. So talking more about population health, concurrent disease histories, and also, of course, the evolution of individual infectious diseases, including we've learned a lot, of course, about plague, but also more recently about hepatitis B virus. I discussed about leprosy, tuberculosis. So, of course, the analysis of these data sets has been very much, I believe, enriched over the last years through very fruitful collaborations between historians, archaeologists, and geneticists. And I think we can all be quite proud about where the field is moving. We're moving much more towards synergistic research directions, not only through the discussions that are taking place, for example, this discussion, but also common publications, but also more recently, conferences that have been organized by colleagues from different fields, joint research grants. So of course, these are all very important directions. Of course, not everything is perfect. I think we have still several obstacles to overcome. And one of the main ones, at least from my perspective, is understanding each other's methodologies in more detail and learning how to better deal with contrasting results. And I think that even within our own fields, we have these challenges. And of course, these are challenges that very much extend when we talk about people that work in different fields and are trained in different ways.
Thank you very much, Maria. Kyle.
Well, I would start by saying that I think we are very fortunate as historians to live at a moment where new technologies and techniques are introducing the possibility of new ways of seeing the past, complementing our traditional ways. And in my view, very much in the early days. In 10 or 12 years, we might think that some of the things we do and say today seem relatively simplistic. And yet, it's amazing how much has happened, how quickly. I forget what year it is. We're 10 or 12 years into the current era of applying essentially high throughput sequencing to archaeological samples. And the development along with that of the necessary bioinformatics apparatus, as well as sort of a domain understanding of how to process and understand archaeological DNA, has really revolutionized what we can know about the evolutionary history of life, of humans, of microbes. And so for historians, it's worth being aware that this is really still barely more than a decade old. And it's very much in development, but it's already made major contributions. I mean, starting with the fact that we are now able, with much greater degrees of confidence, to know what specific pathogens infected specific human beings in the past. And for those of us like you and me who've worried about this for a long time, that's really cool, because there's always doubt working with written sources about what diseases afflicted our ancestors. And even the bubonic plague, which is in some ways one of the more distinctive pathologies identifiable from written sources, people doubted what caused the Black Death. Smart people, informed people. And to be able to move beyond those doubts and to get definitive answers is really exciting. We don't always get that as ancient historians. Ancient historians can debate things for generations. I mean, was the ancient economy primitivist or modernist? Like, there's not going to ever be a lab that tells us the answer to that. So this is kind of new and kind of scary, because sometimes we can be proven right or wrong. 5 years ago, I thought the Antonine Plague was caused by smallpox, the virus that causes modern smallpox. Turns out that's not right. And unfortunately, was wrong. But really, fortunately, we can now know more about the history of this important viral lineage. So it starts with really knowing the specific pathogens that then lets us ask questions about the biology, about the ecology, about the evolution. What are the origins of particular pathogens in time? We're learning a lot of pathogens are younger. They're only hundreds or thousands of years old. These aren't, in many cases, pathogens that have existed for tens of thousands of years. So their evolutionary history is recent, and it's often very active. They move between species, they go extinct, they come back, they adapt. So we really are learning how much we don't know about the evolutionary history, but we're also starting to be able to situate in time and sometimes in space, knowing where pathogens come from. Is it a New World pathogen, an Old World pathogen? Is it from Asia? Is it from Europe? Is it from Africa? How did it move? And increasingly, as we get more and more data, more and more points of insight from specific genomes that can be located in time and space, combined with the family tree or phylogeny of pathogens, we're seeing how they evolve, how they move between species, how they move over space within human societies. It lets us ask questions about the social factors of disease, which are the kinds of things that have always interested historians. And now we can do it in conversation with biology. So we're at an exciting moment where we're learning a lot. We've learned a lot already. But I think over the next 5 or 10 years, we're going to learn even more than we might have hoped for.
Thank you very much. And I think that Maria already brought in something very important. So the follow-up question is for Maria and then Tina: do paleogeneticists ever struggle to understand how historians work? Are historians transparent enough about their methods and arguments for paleogeneticists?
Thank you. I'm actually not entirely sure if this is really a question of transparency, because of course, any field can benefit from being more transparent about their methodology or their methods description. But maybe it really relates to different training or practices in the different fields. And so I can give an example. Within paleogenetics, but in general within any scientific field, we have been trained in a way where each scientific paper that we write, that we read, or that we evaluate has a methods section. And these kind of sections are not often present within historical studies or in the analysis of textual records. So it can be, of course, difficult for us to assess, the validity of an assessment that we read in a paper if we are not familiar with the primary sources ourselves, which we most often are not.
Thank you very much, Maria. Tina, I think you can reply.
Yes. No, I just want to underline everything Maria just said. I am always struck when I read a paper from a scientific journal, how there is always, without fail, a very long section that in great detail describes the methods. And full disclosure, it's often the section I understand the least as an historian because it's often the most technical. But historians are terrible at this, and we tend to not engage in this kind of explication at the beginning of our work the way that scientists are obliged to do, really, because of your publication practices. Some historians will, in introductions to books, discuss their method, discuss perhaps the approach they took, but we're not very self-reflexive in comparison, I would say, to other disciplines. We might describe our method, but we might not then step back and ask, what are the limitations of that method? Like, what are we not telling you by thinking about our evidence in a particular way? So I do think that this whole endeavor of collaboration moving forward, I think historians are going to have to step up and start being a lot clearer in their own work in terms of not only their methods and their approach, but what are the limitations of the choices they're making to begin with in terms of interpreting the evidence. In many ways, and this is the last point I'll make on this, our historical evidence is the equivalent of your sample, right? I mean, it is not, it's raw and it's not interpreted. It doesn't become data until we read it and do something with it. And that process of reading and interpreting needs to be explicated, I think, much more clearly than we are accustomed to doing. So I agree 100%. We need to be much more transparent. And I think that is a good word. We need to be clear.
We can now move to some questions about how data is retrieved, about methodologies, and the explicit or implied limitations and challenges. So the question that I would like to pose to Marcel first. It's not hard for historians to understand the process of extracting material from human remains, but the move from dental pulp or skeletal samples to a sequenced pathogen genome is a lot more difficult to envision. What steps are involved? How long does the process normally take? And how many areas of expertise are typically called upon in the process?
I guess the easy part to understand is that to extract or to cut a bone sample or a tooth sample from a human skeleton, what we then next do is we take it into a clean lab, because all of this work then has to be done in a protected environment to avoid contamination. And the next step is also relatively easy. We would just drill some powder or prepare the sample in another way to mill it down to a powder. And also the DNA extraction itself is rather simple. It's basically a water-based extraction where we add a chemical and an enzyme to basically release the DNA from the substrate. And then we purify the DNA, so we just get rid of all the compounds in this watery solution that we don't need and don't want to have in the end, things like proteins or other biomolecules. The more complicated step is then what we call the library preparation, where we prepare the DNA on the one hand to be amplified, which means we make molecular copies of these small DNA fragments that we have in our solution. And at the same time, we also prepare the DNA by this way for the sequencing, which is then the next step. And then with the shotgun sequencing, maybe for an historian, it's easiest to understand if you compare it to a physical library, like a room full of books, thousands of books. But first of all, you would shred all of them. So you have only like small snippets of paper with a few sentences on it. This is basically the material that we work on. And this is what we get out of the sequencer in the end. And I think the more difficult to understand part really begins there when we have to work with all of these little fragments, all of these sentences. And basically what we then try to do is, in our modern approach with a metagenomic screening, to assign these fragments of pages of a book back to the original book where it's coming from. So we have half a page, we read the sentence, and then we try to understand, is this now a fictional book? Is it non-fictional? Who could be the author of this? And this is basically what a metagenomic assignment then is, which allows us in the end then also to see what books were present in this library originally and which ones we cannot find. And once we know that, we can then go to the next steps. We can specifically fish out the fragments that belong to one book that we are particularly interested in. In this case, in our case, it would be the genome of one particular organism, be it human or be it a bacterial pathogen. And then we can further work with this material. Yeah, this is then basically how I would explain a genome reconstruction. The process to get there, here it's important, I think, to understand that we are working in batches. It wouldn't make sense to do one sample after the other, from sampling, extraction, library preparation to sequencing, but we normally take along something around 20 samples at a time. Also, bigger labs are now using pipetting robots that make it much more efficient time-wise. There's less hands-on work than in the end. And I would say if everything goes well, you could get results within 2 or 3 months, maybe even shorter, if this is all you're doing. But there are a lot of bottlenecks that you can have in the process. I think the most important ones are manpower and space in the lab, because in the end, it's still a lot of manual work that has to be done. And then another thing, depending on the lab also, is queuing for the sequencing, because sequencers are very complex machines, very expensive machines. And most universities have one core facility for sequencing where all the groups from the biological institutes and the medical institutes are submitting their samples for sequencing, and then also the paleogeneticists. And this is often not foreseeable how long you have to wait for the sequencing data in the end. 2 to 3 months, even if everything there goes well, is pretty optimistic, because we have major delays due to multiple reasons. I think the most important one is poor data quality, and that's something that we face all the time with ancient DNA, something that we don't have good workarounds. So here it's often then also a strategic decision how to move forward. Do we want to sequence deeper? Do we want to resample? Do we want to try different protocols that are available for certain problems? But this always basically means that we have to spend more money and more time.
Thank you very much. Tim.
Marcel was also getting at there the importance of collaborations. And I think that is something else that we should emphasize, the importance of setting up collaborations, of building networks of people to work with on different projects, networks of people who are raised in disciplines that are not our own. Setting up collaborations, maintaining collaborations takes a lot of effort and a lot of time. And many collaborations, I, from my own experience, know that they don't always turn out. So a lot of time can be spent invested into projects that are either backlogged or just don't come to fruition. And the collaborations also often entails preparing many applications for large amounts of funding. All of these things, I think, are issues that historians are not accustomed to. They're certainly not trained to navigate. And I think that these are issues that perhaps going forward, graduate students would do well to get some sort of training in. At least this is I'm thinking of a North American context. But even identifying people one could work with is time consuming and often foundational, because I think that collaborations need to begin from the ground floor, that the various representatives of different fields need to be present from the beginning. And to do that, it kind of forces those who are going to engage together to be literate, at least to a degree, in another field. And also, and this can be difficult for historians, being flexible to adapt to the routines and expectations of another field of study, to the schedules and to the publication cultures, the publication expectations of other fields, and being on top of things. That sounds like a weird thing to say maybe, but for all of us, it's not. But the idea that things are in flux and they're constantly changing, and we need to be aware and alert and not check out. We need to be on top of things. And that really brings me back to something I was thinking about when Marcel was talking there. I was wondering if he could reflect upon how the institutions with these labs, the number of institutions with these labs has evolved over the years. Where are paleogenetic studies being conducted? And are there more institutions engaged with this field of research than there were previously? I assume the answer is yes, but I don't know.
I would like now to ask a question first to Marcel and Tina, and Tim can also offer his views. Are there hidden disciplinary idiosyncrasies, traditions, and methodologies that are problematic in collaborative work, or even hinder it in some cases? Marcel.
Thank you. I think there are very practical things that affect the way we are working in our disciplines differently, that emerge from traditions. Some are quite obvious. For example, historians are expected to publish books, which is often demanded by the university to make really an academic career. Books are almost invisible to scientists. It's a bit different, I guess, for us working in an interdisciplinary field, but in a hard science discipline, books don't really play any role anymore in the modern academic world. Instead, we aim to publish in peer-reviewed journals, which, of course, is also rising in history. But then also when it comes to publish in journals, there's the difference between multi-authored and single-authored papers. Historians, I get the feeling, often think that multi-authored papers are not really of value for them. And of course, this is not always a personal opinion, but also depends on the academic structure that they are in. Whereas for us, it's pretty normal to publish papers with 20 authors or sometimes even 100 authors if it's a really large scale project. But I think there are also more subtle differences within the fields. One that's also a personal observation is that historians tend to build their careers more on their hypotheses or ideas. And they very often have to stick to that and defend it against their colleagues criticizing it. And there, I would say that the culture in natural sciences is quite different because if we are contradicted, if there's contradictory evidence, we are very happy to change our opinion. Yeah, and then another point is I think storytelling is something very essential both for historians and for scientists, at least good scientific papers, even if they are not an ancient DNA, they always tell a story. But the way we are telling our stories can be quite different because we are coming from different backgrounds. So especially when it comes to ancient pathogen and diseases in the past, I often hear that historians think that the stories that we are telling through our ancient genomes, okay, we have found plague in the 5th century, can seem quite trivial to historians. But on the other hand, if historians speculate about finding smallpox, evidence for smallpox in the 3rd century, it can be very speculative for us in a way that we wouldn't dare to publish a paper on that. I think there it can sometimes be difficult to find a common ground, really, and to meet each other and really develop a project together and write papers together.
Yeah.
I would very much sort of agree with a lot of the things you just said, Marcel. And well, first of all, I would just say that just to keep in mind that we historians are a varied group, and that I have methodological debates with other historians about how to work with materials. So what I'm about to say, I should probably be clear, is really specific to cultural historians. I think historians, cultural historians in particular, I think you're right. I think we're much more comfortable with speculation, and we're much more comfortable with being not terribly cautious in some cases about the kinds of hypotheses we put out there. And I think that's a positive thing because we're not so much constrained, perhaps, in the way that scientists may be by certain disciplinary requirements, but also in terms of how we produce knowledge is really different. I mean, most historians aren't empiricists. Most of us deal with samples that are in words. And I think the language piece is really important here, because language, if that is the basis of your evidence, language is endlessly referential, right? I mean, language is not a precise medium for conveying what happened in the past or why it happened in the past. And so this is why we can debate, whether we think the ancient economy is primitivist or modern, or because ultimately, if we're relying primarily on written sources, those sources cannot be reduced to a single A or B answer. And so it's interesting when we start injecting the scientific material into historical thought, because as Kyle said earlier on, all of a sudden we actually do have yes or no answers to questions that we never had yes or no answers to. So I think that is a little bit unsettling for historians. But I think the language piece is key. I also think too that, you're right, Marcel, historians tend to cling to certain narratives, whether they're their own narrative or they're somebody else's narrative. And it often takes a lot to get us to sort of give up that narrative. And I think that is, again, a lot about the way we're trained, about how, and the fact that we primarily read long, discursive discussions of whatever topic we're reading, where we're working on, rather than always necessarily going back always to the primary sources. I mean, we are influenced extensively by secondary source material in a way that perhaps scientists are much less so. And so there's always this kind of balance for us between that scholarship, what other historians have said in the past, versus how we're trying to rethink a set of primary sources in the present. So I think that's also probably a pretty significant disciplinary difference between the way we both work. But I would love to hear what, some of the, on this particular question, I don't know whether anybody else wants to chime in. I'd love to hear what some of the other historians think about this really vital question.
Tim, would you like to add something?
Another element here is that historians, and I don't think historians are the only ones doing this, but we tend to privilege and put the most weight in the evidence that we have the most control over. I find that we often fix novel data from the natural sciences to templates that we've made or that we've drafted a long time ago. The narrative of the Justinianic plague, there are multiple narratives of the Justinianic plague, of course, and first pandemic plague, but there are commonalities that stretch back long before the advent of paleogenetics or even the isolation of Yersinia pestis in 1894. And when we discover, well, not we, but when Maria and Marcel recover a Yersinia pestis genome that dates to the late antique period, I think there's the impulse to somehow find its place or to fit it to what we've been saying within the field of history, rather than us trying to fit the historical narrative or the established narrative to the genome. And I think it even goes deeper than that, because I think, for instance, we look for Yersinia pestis in late antique remains because of the fact that historians say Yersinia pestis really mattered in the late antique period. Kyle was part of an amazing paper published in 2020 on the measles virus, and measles gets no traction. I don't know of any other paper published since on the antiquity of measles involving a scholar in the humanities. Plague is what matters, and I think that's in part because historians have been saying for a long time that plague really matters, without always being, as Tina said before, and I made note of this, very self-reflexive. So I think that's a little bit of a hidden disciplinary tradition or methodology, that when something new appears, no matter how unexpected it might be, the initial reaction is to somehow fix it to the narrative that we have. And, there's a great example here with Edicts Hill. We don't need to get into fine details here, of course, but the impulse to want Edicts Hill to date to after 541 or 542, the discovery of, or one of the Yersinia pestis genomes there that I believe, though Marcel can correct me, was radiocarbon dated to something like 415 to 541. But the idea that it has to date to after 541, I think it displays how historians and many in the humanities privilege the written sources and the established narrative over other novel data, which could shake things up. And as was said in response to an earlier question, one of the things that makes the interdisciplinary study of disease history so much fun today is because it's so dynamic and fast moving. And I think in order to go with the flow, we need to be prepared to abandon preconceived ideas about what happened in the past.
I would like to take on something that Tim just mentioned. I think there's a difference because we as historians, at least I think, we are expected to offer grand narratives, much more sweeping narratives. We're not talking about something very specific and very small, but we're trying to fit it within narratives as in, when did late antiquity begin and end? What caused it? What caused change? What made, the big sort of driving forces of history, the wheels to move? And for that, yes, we do need to formulate a lot of hypotheses, and very often they are quite daring, and very often they come to nothing. But I do think that it's because some historians in the past came up with big ideas that couldn't, at least at the time, be supported with evidence from the hard sciences, that perhaps the hard sciences started to look into other questions and delivered then evidence that either supported or negated some of these bigger questions. So it might also be a question of scale. We are trying, I think, to offer something, the bigger picture. And in order to do that, when we're dealing with the ancient, with the pre-modern world, where data is not always abundant, we have to revert to hypotheses. Let us also not forget that because we're dealing with texts, and texts are also literature, that there is a kind of, literary aspect to our prose, to the way that we write, to the way that we're supposed to write. And that might feel strange, I think, for those writing scientific papers, but it is very important to us. I assume this is not a question, but just a kind of defense, I guess, of an apologia of what we're doing in order to reach, I think, a better understanding. But Tina has something to add.
If I could just interject and maybe sort of there was modification question back at either Maria or Marcel. I'd love to hear this from one of the scientists. I mean, your point about scale, I think, is so important, Dionysios. And I would like to, I'm wondering, I mean, how comfortable are scientists at scaling up, as it were, conceptually, right? I mean, obviously, we can talk about scaling up in terms of producing more data. But I mean this in a more sort of abstract way. I mean, how comfortable are you with sort of taking what data you do produce and using them to say something much bigger than what the sort of, the immediate reference of the data? If that's clear, I'm curious about how comfortable you are with scaling up, because I feel like this is another potential sort of disciplinary barrier between the way we think about doing research that might be interesting to people to hear.
From my perspective, I think that in general, there's a consensus that extraordinary claims need extraordinary evidence in ancient DNA. And I think maybe there are sometimes papers that come with this kind of extraordinary claims with very little evidence have been sometimes published in very high impact journals, but have come also with a lot of criticism from the field. And so I think perhaps one of the main differences here is that these kind of bigger scale kind of interpretations are more used for perhaps review papers rather than for kind of primary research papers within the field. But very happy to also hear what Marcel has to add to that.
People working on ancient DNA, they have very various ideas of how they can scale up and feel comfortable in using grand narratives. There is certainly a tendency to get high impact publications by using exciting but scarce evidence and putting it in a very grand narrative. But sometimes it seems very forced upon the data. And this is maybe also not always clear to the reader, especially from different disciplines. And I think what's also important is to keep in mind the limitations that we have. I think we already talked about this a bit. For us paleogeneticists, we can only work on what we have, which means samples, which is a limitation that's not always clear. So sometimes I also get asked, why don't you work on China? Why don't you try to find plague in China? Well, because it's very difficult, first of all, to get access to the material, which is, I guess, currently impossible. And on the other hand, I guess historians also have their limitations. I can think of language, for example. Not every historian can work on the history of smallpox in China, because I guess you need to speak and read different Asian languages for that.
Thank you very much. I would like now to raise a question that goes behind the scenes to explore important issues that are not always explicitly stated in our publications. So my question to Marcel and to Kyle: when paleogeneticists and historians begin new disease-related projects, they might start with different questions and goals in mind. What are the typical big questions paleogeneticists ask and hope to answer? What do we all want plague to do in our research, in our specific paper, in our concepts? Marcel.
I think to some degree it depends on the researcher's background and also the background of the group. We come from different disciplines. Ultimately, some of us are trained as evolutionary biologists. Others are initially trained as anthropologists. So the very obvious first research direction ancient pathogen genomics went to was identifying the causative agents of certain diseases that we knew from historical accounts or that were associated with mass mortalities in the past that archaeologists identified. But it very fast moved away. And now I think the most important research questions we really want to address things like the time and place of emergence of pathogens, the evolution of pathogens, how they evolved, for example, to adapt to specific hosts, be it animals or humans. Also in a broader sense, what anthropogenic or environmental factors contributed to the spread of diseases, to spillover events from animals to humans, for example. Having all of that said, I think there is generally the trend towards diachronic sampling in ancient pathogen genomics. So we really want to cover larger periods of time, be it centuries or even millennia, where we can really trace the evolution of a pathogen over a long period. So we are talking here really of different scales, because historians, even if they have a longue durée approach, it would be a few centuries maximum, right? So here, I think we really sometimes have different approaches that diverge, and that make it difficult sometimes also to publish the results, because we might publish a paper with genomes spanning several millennia, and a historian who was involved in the project on some of the samples might have difficulties to fit in his narrative there or his interpretation of the results. In the end, I think what also plays a role is that paleogeneticists are really opportunistic explorers and are very data-driven. As I said, we have to work with the material that we have. We have our samples, we try to find pathogens there, but in the end, if we find hepatitis B, we will work on hepatitis B. And we cannot work on plague if we can't find it in the samples, right? So I guess that also sometimes sets then limitations to collaborative work.
Thank you very much. Kyle.
A couple of thoughts. I mean, one, I think we should say clearly something that maybe hasn't been surfaced enough in the conversation, which is that at minimum, this kind of work is triangulating between people who have expertise in genetics, in the biology, who are often very comfortable in the lab, with historians who know the written and documentary record, and have the expertise in interpreting the human side, the social side. But we haven't really, I feel, underscored the importance of collaborating with archaeology too, as a freestanding discipline that brings its own techniques and questions and methodologies, and is crucially important in triangulating this new kind of data and understanding how it's sampled, the taphonomy, the specifics of the symmetry and the context. So really the big questions that we can and should be asking are triangulating between these three different fields. And I would say too that none of them are to be privileged. That I'll stand up a little bit for the autonomous importance of the written record, as complicated and as mediated as it is. We can learn a lot from ongoing critical study of the written texts, and then use that knowledge as well as the walls that we hit, the limits of that knowledge, to work collaboratively to be able to design research that addresses questions. And this, I think, builds on something that I think it was Tim said earlier, that it's very important that collaboration really starts at the beginning, and that that historians and archaeologists and paleogeneticists are working together to design research, instead of in the early days it's about finding samples and seeing what can be done. But as this sort of collaborative field matures, we can work more to design research projects from the ground up, that are aware of the contributions and the limits of each of the respective disciplines that are involved. And so to give an example that's close to me, having worked, for example, on all of the written evidence that there possibly can be found for the plague in late antique and early medieval Italy, there, at the end of an investigation like that, just remain huge gaps. We don't know. I don't know if the plague kind of disappears in the 7th century or if we just don't have enough documents to really see it. We don't really know where the plague is coming from. Is it local? Is it distant? Is it moving north, south to north? Is it coming from the mountains? Is it coming from the sea? And these are kind of questions that when you start a project in conversation with archaeogeneticists, can then work together to think about what's potentially answerable, what are the most answerable questions, what kind of samples would be needed to answer these kind of big questions. And I think that's where the field is going and can go in the next decade.
Thank you very much. Tina.
One of the questions that really interests me as an historian that rarely appears certainly not in the scientific journal version of the project, but perhaps even not as much as one would expect in the historical publication version of the project, which is where do we find a place in this triangulation? And I think that was very articulately put, and I think you're absolutely right. Archaeologists play a huge role in this, and we need their voices to be heard as well. What place is there for the perceptions and explanations of these major disease events like plague that of our historical actors themselves? In other words, what place does Gregory of Tours' understanding of plague, apart from where he thinks it shows up, where he thinks it goes, how many people he thinks it kills, and we can even ask this of any disease, what place do the sort of late antique and early medieval explanatory frameworks, what place do they have in this triangulation, in this relationship between the three? Where do we publish that data, as it were?
I would like to add something to that, which I would very much like, everyone, but especially our previous speakers to address. I think from something that Marcel mentioned earlier, if I interpreted it correctly, you said, if we find plague, we deal with plague. If we find something else, and so the question is, when you embark on research, when we embark on research, do we need a specific answer, a specific role for this pathogen to play in our reconstruction of the past? Are we ready to be surprised, to have to reframe, rephrase, change? I think that chimes in with what Tina said, because there are things that we need to then select and perhaps leave aside. And where does all this material or this data go? Marcel, but Kyle also, of course.
It's a good question. And I think it's good to raise here another point, especially in these interdisciplinary teams with historians and paleogeneticists. The historians involved in these projects, they are interested in disease. And most of historians with an interest in disease want to find diseases in the past, right? There is only a minority who rather wants to make claims that there wasn't a certain disease. So what I observe sometimes also with myself is this tendency towards interdisciplinary circular arguments. So we just tend to confirm each other. And I think that also comes back to a point that Tim earlier made. We find a genome that roughly falls in the right time frame, and the historian says, oh, of course, that's the disease that this chronicler described. And then we as geneticists say, oh, great, now we have a historical framework, we have a narrative to tell. And with all of that, we tend to dismiss all of the data that's not really confirming this narrative or this association. And I think that's also one of the problems that I, in the recent years, had with this idea of consilience that some historians advertised in this interdisciplinary work. Because in a consilient approach, you only really look at the data that's supporting each other. You only see the convergence of evidence, but not really where the evidence disagrees.
Thank you, Kyle.
Well, I'll go back to Tina's question, which I think is a really hard one. And I don't think there are easy answers, but historians and humanities scholars will and need to continue publishing in our conventional journals, but to do so in ways that are aware of the dynamic insights and knowledge that's coming from other fields. It's very hard to do for lots of reasons, right down to the methods, the logistics. I mean, it takes years in many cases. It's unforgivable, but it takes years for a paper that gets accepted to actually appear. I'm not going to name any specific. That's an issue that I feel very strongly in a personal way. And it's absurd because the natural sciences, of course, have much faster pace. The review times are much shorter. By the way, humanities should absolutely adopt like the 2-week review window. You're going to read it at some point, just do it. But the challenges are enormous. And on the other side, looking at it from the more natural science discipline perspective, we need to find ways to work together better to make sure that the supplementary material is of the highest quality and is truly integral to the project, not an afterthought, but that is something that the people have the right incentives and rewards to invest in. A proper archaeology supplement is absolutely essential to interpret, whether it's human DNA or pathogen DNA. You don't really know each individual burial context. You don't even begin to have the information you're going to need to interpret the results. And yet, as we all know, there are a lot of supplementary materials that are incomplete at best. And I think there's, like I say, there's not an easy answer, but finding with a way of recognizing how important that is intellectually to the contribution, finding the right ways to incentivize and reward teams that are doing that right and doing it well, because that's and often those parts are largely written by the historians and archaeologists who know that whether it's a tenured committee or whatever isn't going to really evaluate well how much work and how serious an intellectual contribution that is. That's a big problem, and I think it's getting better, but it's an issue where I think we would benefit from finding ways to recognize that we would all benefit from that being a bigger part of the entire process.
Thank you very much. Tim.
If I could just jump in and emphasize that point, that if the discipline of history, and I guess possibly to a lesser extent archaeology, was to recognize and maybe even begin to encourage historians to participate in these large collaborations, and not only to recognize the amount of time that goes into carrying out a project until it's written up, until it's gone through peer review and published, but also the applying for funding for projects. I don't think many history departments evaluate either applications for funding, either if they've been successful or not successful. And sometimes those applications take months to put together, and sometimes the application is more polished than a publication, but no one reads it. I also wanted to come back to something that Marcel said before earlier, that a lot of historians who work on disease not only want paleogeneticists to find disease, but they also want the disease to matter, of course. There's only a smaller subset maybe that is less concerned with whether or not a disease mattered in the past. And I think that this just ties back into just how influential and not sufficiently interrogated the role of history has been in the pursuit of earlier paleogenetic work. The one thing that struck me about the 2019 paper that Marcel and Maria led was that there were samples taken from sites that didn't line up with the chronology of the Justinianic plague, which was wonderful. But then it made me think that what would you have done had you found Yersinia pestis in the 300s, or essentially if you had found it when you weren't supposed to find it? As a scholar who spent a lot of time reading and thinking about late antiquity, I think that could be remedied with greater ease than if you were to find Yersinia pestis, for instance, at a number of sites in 11th century France, or some point in the High Middle Ages when agriculture is expanding and population is booming. But I don't know where I'm going with this, but I just think that so far Yersinia pestis has fallen into time and space where it can be accounted for, and where the expectations that we tend to have of Yersinia pestis can be met. But if it was found in another region or another period where we wouldn't expect it, or where historians wouldn't possibly want to see it, then I don't or know how to explain it, then I think that would be quite an interesting thing to behold. And maybe that will happen as the search for pathogens expands, as you were both just saying, and becomes less fixated on big narratives.
Thank you very much. And that brings us, I think, beautifully, organically, to our concluding question that is first addressed to Kyle and Maria, but I think we could all offer our views on this. What is on the near horizon and the more distant future regarding pathogen, paleogenetics, and disease history? Maria.
Thank you. Yeah, I mean, I think we can all agree that we have to continue working together. So this kind of intense spirit of collaboration between different fields must continue, and must not only continue through publications, but as several of you already said before, through joint research projects and through discussions that start at the beginning of the projects, for example, through formulation of research grants. And this is something that has already been happening. Other than that, I think it's really important in terms of kind of scientific progress to make more and more efforts to escape sort of the Eurocentric views that a lot of us have in terms of disease history. This is something that I personally also would really like to focus on, continue working on in the future. So investigating more contexts from different perspectives in Asia or even Africa, both in terms of plague history, but also different kind of diseases. And also from a methodological perspective, I also think that there's actually still quite few pathogens that have been studied that we have focused on over the last year. So really expanding the array of pathogens that people work on, and not only kind of cherry picking the diseases that we think are important, because we find stuff and we often maybe neglect or put them away and kind of focus more on the sort of more deadly kind of diseases that might make more impactful stories. But also looking more at epidemics that have not been analyzed so far. Like a personal interest, a high interest of mine is the plague of Athens, for example. So there's still also a lot of several epidemic or pandemic events that have not been investigated so far, or that we have not been able to yield meaningful results from so far. And I think those can be definitely looked into, with the development of more methodologies, including, for example, the analysis of RNA that has been also discussed in this discussion. Of course, from archaeological context, this is going to be much more difficult than what has been done already. That is mainly from medical collections, but it would be a fantastic sort of endeavor for the more distant future.
Kyle.
Well, Maria said it extremely well. But I think as this interdisciplinary field continues to mature, as there are as the methodologies kind of have become standardized, as there's more labs doing the work, it opens a lot of opportunity for broadening collaboration with historians and archaeologists to design research projects together that are asking questions in a collaborative way, that are sampling and analyzing in ways that are driven by gaps in our knowledge. So I think we'll get more data, more spatial representation in the data. We'll get more micro studies of in space and time. We'll get a broader sense of diseases, of their movement between humans and animals. We need more sampling of animals and their pathogens to really understand the microevolution and history of these pathogens in recent time. So we're going to be able to ask new questions, hopefully, as we really continue to fill in little points in the map on phylogenies. We're able to ask questions about how diseases move, what are the social factors, what's the role of hunger and biological stress, of co-infection in shaping human health. So the research agenda for the next 5 to 10 years is hopefully driven by new questions that go beyond just the identification of pathogens, but really start to deepen the questions that we can ask about the human experience of disease.
Thank you very much. And I'd like to open the floor, so to speak. Are there things you wanted to add? Is there something that in the course of the discussion came up and you think we can revisit, we can mention here, we can explore? Tina.
So I really just want to add on to how we might envision this collaborative future, and add a couple of other thoughts, which is, well, I definitely think that history departments and historians need to become more familiar with the models of collaborative research that have been normative in the sciences in North America for a very long time, but are much less normative in humanities departments. In Europe, this is much more common. You see these interdisciplinary research projects that get these huge funding packages, and we just don't operate like that in the US. And so I think historians need to start looking to Europe more for models, but also looking to our our neighbors in the physics department, as it were. But I also think that the scientists have to also perhaps get a little more comfortable with doing things the way historians like to do them. And I want to plug the book again, because I know that the scientists don't read books anymore, don't write them anymore. I mean, you read them. I don't want to say you don't read them, but you don't write them anymore. I actually wonder whether the book is in some ways a solution to the problem of how do we think about these disease events / developments at different scales that incorporate different perspectives, not only the perspectives of us, the scientists, the modern historians, but also the perspectives of the people who were actually experiencing these disease events. And I wonder whether there's not going to be enough space in an article in any kind of journal really to incorporate all of these things. It might also be a book as a place where there's space to address the shortcomings of a consilient model, that is, to really take seriously the divergence and disagreement amongst our data sets and our interpretation of data sets. And so I wonder whether this kind of new frontier of collaboration also has to think about maybe older ways of doing things as another possibility. I also think if you said that, more historians would probably jump on for a variety of reasons. And then I want to make one short plug for a project that Kyle Harper is currently working on, and I want to hear, hopefully he can tell us more about its publication, which is a new translation and an edition of a text that has been really crucial to the study of late antique plague, and that is John of Ephesus's plague narrative. And this is a really amazing source that we don't use well enough or nearly as enough, in part because our translations are problematic for various ways. And so I want to just hats off to you, Kyle, and your co-author and / translator, Scott Johnson, on that one. And I'm looking forward to reading that. And I want to encourage more historians and linguists to dig in and revisit these texts that we think we know, because I actually don't think we probably know them nearly as well as we need to.
Yeah, thanks. And I think the deeper point is just that the arrival of archaeogenetics really only increases the need for humanities scholars to do what we do really well, to really go back to texts, to ask questions, to dig into the language. And I think one of the cool things is you're seeing that in our field, people are going back, people are digging harder, people are looking again at things that maybe we thought were familiar. So there's far from being some kind of antagonism between the more bioscience approach and the more philological or traditional approach. There's a lot of opportunity for collaboration, but also just simply to do what the kinds of things we've always done, but maybe informed by new questions and new data, but still in a very, very honestly conventional mode of looking at texts and trying to understand what they mean.
Tim.
I would also jump in to say that as we move forward, I think including, of course, archaeologists in this discussion, but also paleoclimatologists to help us untangle how disease experiences in the past, epidemics, spillovers, and also the canceling out of outbreaks of disease could have been impacted by climate change. Historians have been willing to speculate on these sorts of things, but I think we need more data yet to answer those sorts of questions, not only from paleogeneticists, but also from paleoclimatologists and scholars who are invested in the reconstruction of the landscape, historical landscapes, like palynologists.
Just to follow up on Tim, I think that's a good point, and also points that Kyle earlier raised. I think the tendency really will be towards more interdisciplinary studies involving more and more specialists from diverse fields, and also what we haven't explicitly talked about, but indirectly, Kyle, for example, physical anthropologists, really to use the skeletons as archives of health and disease, and to use the data that we have available there, for example, with a paleodemography. I think only by integrating these lines of evidence, we can tackle these genuine historical questions, for example, about the impact of diseases on local populations.
And really, there's so much opportunity with that. If we expand the teams, if they expand the number of disciplines typically represented in an interdisciplinary team, and if there are more teams operating, we'll learn so much about the disease experience of the late antique period. Thinking, for instance, just of the Justinianic plague, just of the initial wave of the first pandemic. If we are able, with the help of paleogeneticists, with palynologists, paleoclimatologists, to undertake a number of local case studies where we can discover, find, recover Yersinia pestis spreading through different environments at different times of the year, at different points of time between 541 and 544, maybe before and after, through landscapes that were malarial, through landscapes where tuberculosis, from the pathogen's perspective, was doing well for itself. I think that we would just stand to really enhance our current interpretations and understanding of major disease events in the antique period.
Thank you, Tim. That's half a research project there. It's almost a bid. Maria.
Just following up again on what Tim said, I think it's also very much gives perhaps a new perspective on what we were discussing before about these kind of broad strokes and big claims, and rather emphasizing the importance of perhaps more detailed studies, or more focused studies on these different aspects that could have a sort of wider impact in terms of the disease history as a whole.
At the same time, I think it is important to note that in order to find something, you have to begin with a hypothesis, with a question, and that already points you in one direction or in some directions. Revisiting material, we're not completely, clean slates, blank canvases. We are already looking for something. And the fact that we are, of course, reading texts that need to be translated, even if that it's in our mind and it's happening automatically, we're already going in various directions. The sort of micro level is very important, but I think we do also need the bigger picture. So the micro studies need to fit and to feed into ideas about reconstruction of a research question, specific historical periods and the forces that shape them. So I think we need, both zoom in and the sort of, taking several steps back and looking at our material anew. I think that the discussion offered us, I can speak for myself, very many very important new insights, and I could understand certain things much more clearly, taking notes that, for when we go back to our own more solitary desks and tasks. I would like to thank everyone involved very much, especially, of course, Tina and Tim for conceptualizing this, coming up with this great idea. And I'd like to thank everybody for their time and for their frankness and for their willingness to engage in this discussion. And I hope that our readers and listeners will profit as much as we did from this discussion. So thank you all very, very much.